Hi guys, welcome to my presentation today. My name is Eric Okoyo. I am a trainer at the Eldoret National Polytechnic Department of Health Science. In my today's video, I want to take you through a brief introduction to a test that is most commonly performed in a blood transfusion science laboratory, and this is called the antiglobulin test. Now, antiglobulin test is also referred to as the Coombs test. So basically, I'll take you through the principles and practice of antiglobulin test or Coombs test. Now, basically, antiglobulin test is a test that aids in the detection of IgG antibodies. Why do we emphasize the detection of these IgG antibodies? When IgG antibodies take part in reactions, they don't produce a visible result. There is no visible result of this reaction. So reaction has taken place, but we cannot see that this reaction has taken place. So sometimes you can mistake it for a negative result, all right, where there is no reaction. Okay, so IgG antibodies will sensitize red cells either in vivo, or the red cells can be sensitized in vitro. When we talk about in vivo sensitization, we mean that the red blood cells are sensitized within the peripheral circulation. In vitro sensitization, on the other hand, refers to the sensitization where these red cells are, sensitized with the antibodies on the bench when we are performing our routine antigen antibody test. So Coombs test will enable us to detect red cells that are either in vivo sensitized, or we can sensitize our cells in vitro and then we check for sensitization. And that will bring us to the two types of Coombs test that we have, which is direct Coombs test and indirect Coombs test. So when we are detecting in vivo red cell sensitization, we shall perform what is called a Coombs test. We are going to see the procedure shortly, in the subsequent slides. And when we are detecting in vitro red cell sensitization, then we are basically performing what is called an indirect Coombs test. So what is the test principle? The key thing about this Coombs test, is based on how IgG antibodies, take part in reactions. Now, because of their small size, when they take part in reactions, they bind to the corresponding, cell-bound antigens, but do not agglutinate those red cells. Why? Because their small sizes are very small. So due to their small sizes, they will take part in a reaction, but they are not able to bring about agglutination. Now, agglutination is one of the results of a reaction. So whenever you see agglutination, then that would be a confirmation that a reaction has taken place. So if an antibody takes part in a reaction and you're not able to see agglutination, you can easily confuse that there's no reaction. So the use of anti-human globulin in this case, which is our Coombs reagent, enables agglutination of the red blood cells. Okay, so the anti-human globulin, this anti-human globulin, which is our Coombs reagent, is actually antibodies that are prepared, the antibodies that are specific to the human globulins. That is why we call anti-human globulin. So the antibodies that are specific to the human globulins. So we can easily purify human globulins, IgG in that case. Then we introduce into laboratory animals, and then the immune system of these animals will respond to these foreign substance. In this case, they will produce antibodies against these human antibodies. Okay, and therefore we are producing the anti-human globulin. Now this anti-human globulin now becomes specific to the human globulins. So whenever they interact with the human globulins, in this case our IgG, then they will react with the human globulins because they're specific to it. So the anti-human globulin, being specific to the human globulins, binds to and bridges the gap between any two neighboring IgG antibodies that have sensitized the red blood cells. So what we are saying is that when sensitization takes place, the IgG will have bound the antigens on the red cell surface. Now, anti-human globulin now binds to these antibodies to be able to bring about agglutination. Okay. Now, there are two types, and we have said the first one is direct Coombs test, and the second one is the indirect Coombs test. So, direct Coombs test basically detects those antibodies that are sensitized in vivo. Okay, so sensitization has taken place in vivo. So the suspected red cells are basically washed and the Coombs reagent added, and then the sensitization is checked. So when you add the Coombs reagent, a reaction or agglutination will be a positive, test, and lack of agglutination will be a negative test. And as we have said, washing is very, very, very key before you add our anti-human globulin, because if you don't wash, then you're going to come up with false positive results. The last one is the indirect Coombs test, which now detects the presence of IgG in serum. Okay, so what we are saying is that we have serum that is suspected to be containing IgG antibodies. We react this serum with red blood cells that contain antigens corresponding to the suspected IgG antibodies. So if the antibodies are present in the serum, then the added red cells will be sensitized. Now, this is called in vitro sensitization. Then we will wash, and then we add our anti-human globulin to check for sensitization. That marks the end of my presentation. So I hope this video helps you. Please subscribe, and welcome to our next video. Thank you.